Yep.
Cool.
That's all you need to know.
That's all I need to know. I'll find out all of the other things about you.
Yeah.
In a moment. Well, hi, welcome back to another episode of Explain Yourself. On my erstwhile adventures in Austin this spring, I still managed to connect with my friend Erik Iverson. He's a PhD student at UT Austin, so we're going to learn more about him and his research and stuff today. So thanks for being on the show.
Of course, happy to be here.
Awesome. So the first thing I always like to ask people is, could you please explain to us your current research?
Yes, so I work in a lab called the Harvard Lab at UT Austin, where our focus is on the mitochondria. So a lot of people know that mitochondria are the powerhouse of the cell, quote unquote. They are involved in creating energy for everything that cells need to do, and so all eukaryotes, which includes all animals, all plants, all fungi, have mitochondria in every one of their cells. Our lab studies what mitochondria do, how they influence the evolution of organisms or the ecological niches that they can live in, and we're particularly interested in the genes that mitochondria contain. Some people may know that mitochondria have their own genome, distinct from the DNA in the nucleus of a cell. And they have a small number of genes that are involved in these core energetic functions that they perform. So our lab studies the function of those genes and the interaction of those genes with nuclear genes. So there's interactions between these two different sources of DNA in the cell. But those two different sources of DNA evolve in different ways at different rates and for different functions. So there can be conflict between them, there can be coevolution between them. So we study the ways that organisms adapt through both of their genomes, what happens when DNA is in conflict or when it's working together, and sort of the downstream consequences of that. So where I come in on that is my background is more in behavioral ecology, more working in an environmental setting. Often I've studied birds in the environment, what they do in their niches, and how they interact with other birds. And I have become interested in some theories about how the mitochondria of birds should be involved in sexual signaling, in processes of hybridization between birds. And so I'm going to be working this summer on a hybrid zone here in Texas, where two species of titmice are coming together and breeding, actually very close to Austin. And so I want to see if there is any signal of basically conflict between mitochondrial and nuclear genes in the hybrids, where you've got genes that have never had to work together before, working in the same organism. Do we see any reduced fitness in those animals? That's kind of the question I'm pursuing.
Wow, that's really cool. And so when you started talking about hybrids and mitochondrial versus nuclear DNA, immediately my mind went to the fact that mitochondrial DNA only is contributed by the mother of the organism. So yeah, what do you think about how that's going to play into this idea of hybridization, where like mom is one species of titmouse and dad is a different species of titmouse?
Yeah, you get situations where you have different combinations, right? So you may have the mitochondria coming from one parent or the other parent. And what we see in some other hybrid zones is we can see if one parent ends up more often contributing mitochondria to the hybrids, maybe because females disperse further or something, we can start to see the nuclear genes become biased towards the parental species that contributed the mitochondria. And that's, we assume, because over time, due to the need for coevolution and coadaptation between these genes, if you've got mitochondria from one species and a nuclear mismatch, those nuclear genes are recombining, they're getting shuffled down the generations and selected for or against, and over time selection will retain the nuclear genes that came from the same species as the mitochondria.
Are there specific gene combinations or like specific gene functions that you're most interested in?
Yeah, so the genes we're most interested in are the genes of the electron transport chain. So the way a mitochondria generates, or a mitochondria on a single one, generates energy is through a process called oxidative phosphorylation. The way this process works, and we're getting back into like basic biochem.
Yeah.
I actually had to relearn all of this stuff because I hadn't dealt with it since AP biology.
Right, well, and the difference between like what you get in like high school or like undergrad biology versus like the real deep dive details that you guys are working on is like a completely different level of understanding, right?
Yeah, exactly. But I tutored a student about a year ago in his like intro bio course, and I was like, I don't remember any of this, but it's good that I relearned it because it's what we do now. So mitochondria generate their energy through oxidative phosphorylation, and what that means is basically they are generating a gradient of hydrogen atoms, protons, and then using that gradient to do work. So kind of like water held back behind a dam, they're selectively letting electrons flow through, and it's literally spinning a physical turbine, which is an enzyme called ATP synthase, and that enzyme phosphorylates ADP to ATP and makes that cellular energy currency we all know and love. So the genes we're interested in are the electron transport chain complexes, sometimes called cytochromes or respirisomes, and basically these are proteins embedded in the mitochondrial membrane, and when an electron enters the system from the food that we eat, having been broken down at an earlier step, it then flows through a series of proteins, and as it gives some of its energy to each one of these proteins, it causes a conformational change, which pushes a proton across the mitochondrial membrane and generates that gradient, and then at the end they all fall through the gradient. And so
it's like flipping a switch at each point?
It's almost like if you had two different liquids and you were using another one to pump water to eventually get your energy flow. So you were like driving oil through a hydraulic piston to pump water and then taking the water and letting it flow downhill.
Okay.
And so those electron transport chain proteins are often what we call chimeric, which means that they are composed of pieces from two different genomes. So this is where you get that physical interaction between mitochondrial and nuclear genes. For instance, if we take complex 1, you have a core set of proteins encoded by the mitochondria, and then nuclear proteins come and wrap around and complex with it, and you can't make this without components of both genomes. 3 of the 4 proton pumping protein complexes work like this. They're made of pieces from both the mitochondrial and nuclear genomes. So we're interested in those genes, and we know where they are in both genomes. There are hundreds of other nuclear genes that interact in this process, helping these proteins form or being involved in getting electrons to the system, influencing the state of the system. But these are the core sort of mechanistic proteins. And what we're also interested in is not just the entire protein, but the sites where they physically touch. And a lot of work has demonstrated when proteins interact, you can see the signature of natural selection at one protein and then in another at those sites where they actually interact. So we expect that one protein is sort of being selected along with another, and there's a hypothesis of what we call nuclear compensation, the idea that the mitochondrial genome is mutating faster and with more error. And so nuclear genes are playing catch up to the mitochondrial genome. They're being selected to change their conformation to match the conformation that changed in a mitochondrial genome.
Interesting. So this is a very intimate relationship between the nuclear genes and the mitochondrial genes in this process that you just described. So yeah, if the nuclear and mitochondrial genes don't play nicely together, it's like disastrous for the organism, right?
Yeah, it can be absolutely fatal. So there may be in situations where proteins come together that are not well adapted, like in hybrid zones, maybe you never get offspring in the first place. And this is something we see, right? There's a lot of hybrid inviability or species that just can't even produce offspring. Maybe they produce zygotes, but all those zygotes die. This could be one mechanism. Even in the zygotes that implant in humans and other species, a lot of them never really make it, right? A lot of them die after implantation.
Right.
It could be because there were incompatibilities here or in other genes, but we think that these mitonuclear incompatibilities are a big source of the incompatibilities we see in hybrid zones in general. And there's been a lot of questioning over the years as to what the actual mechanistic basis of this hybrid incompatibility is. And so we think that we might be onto something here.
Okay, so is that the sort of like, so what about this research? Like, what are you hoping this research will do in terms of information or like change making? Yeah,
so that is one so what. That's one take home, is that we have been wondering for, over 100 years about what prevents hybridization between species, what enforces the boundaries between species. And so my research, part of it is directed at testing this hypothesis that when populations are in isolation from each other, their mitochondrial and nuclear genes co-evolve with each other, but down different pathways. And when they come into secondary contact, there may be incompatibilities between nuclear and mitochondrial genes that enforce species boundaries. So that's one key thing that I want to test, and I think that has a lot of implications for evolutionary theory in general. Now, there are also more ecological implications. So this research I'm going to be doing with titmice involves two species that are adapted to different climates. So in the eastern half of the range, basically east of Austin and throughout the rest of the southeast, we have the tufted titmouse, and west of here we have the black-crested titmouse, which is adapted to these drier habitats in the Texas hill country, more of an open, brushy savanna than the sort of closed, wet forests you get throughout the southeast. And so I've been wondering if these birds are adapted to different climates and whether, rather than just, through the process of genetic drift becoming different from each other, whether they have also acquired adaptive differences in mitochondrial function that might contribute to incompatibility. So we see in some species that, there's long been this idea that there's not much adaptive variation in mitochondria because they're so important. All that variation is going to be selected out through purifying selection, that they will basically have a uniform genome. But there's actually a lot of variation there, and increasingly people are linking mitochondrial genetic variation to the environment. And big climatic factors are probably the main driver of differences there. Temperature, rainfall, food availability derived from rainfall, altitude. So those kind of things. And so I'm interested in seeing whether any differences between that we see between the species are related to the climate, doing correlations between the genetics we see in one place and the values of mitochondrial respiration we observe, like how well mitochondria function, seeing if that's related to local climate at all, and whether differential adaptation can explain differences and incompatibilities between them.
Wow. That's cool.
You can imagine if you've got a situation where we're starting to understand the mechanistic basis of why species are adapted to particular climates, this has implications for our changing climate. So I would love to know whether we can predict changes in the ranges of these two species, changes in the frequency of hybridization or the consequences of hybridization. One thing we see with hybrids sometimes, or with any genes, honestly, with any genetic interaction between genes, is that it may be fine under one environmental circumstance and not so fine in another. So you might have genes that when times are good, they work well enough to get by. But when times are bad and the organism is stressed, you start to see them failing. So maybe these hybrids don't display lower fitness under normal conditions, but what if it gets 5 degrees hotter?
Yeah, wow. So yeah, it might sound like a pure science question, but the implications are really far-reaching and very relevant right now.
The implications are pretty huge, and there are also a lot of implications of this kind of research for human health. So we have identified mitonuclear incompatibilities in human populations. It's not so obvious that it's anything like there are two populations of humans who shouldn't mix because they're incompatible. It's not that simple, but there are genes out there in human populations that don't play well together, basically. And we are starting to identify what those are. There are a host of mitochondrial diseases that cause impaired function, and we are seeing that some of them are caused by the interaction between mitochondrial and nuclear genes. And if you're a carrier for a deleterious mitochondrial mutation, you might not express it if you have the right nuclear background. Or you might express it in the wrong nuclear background. So we're learning about that, and any information we can gain about what these genes actually do on a physical level can help inform human health.
It's amazing. That's really cool stuff. So I guess I also like to sort of take it back with people. So you're working on genetic stuff and birds, but why genetic stuff in birds? Why a PhD? Why become a scientist in the first place? So tell us how you got to here.
Yeah, so I guess it goes back really just to a love of nature and especially of being outdoors, which is ironic because now I'm being in the lab more and doing a lot more computer type stuff with this research. But I'm very lucky to have had a family that wanted to go outdoors for family vacations, do road trips, go camping, things like that. I was in the Boy Scouts, and so I got to do a lot of camping, and I just loved being outside and started to learn more about the animals there, and sort of teach myself basic natural history. And when I got to college, I really got into it through classes I took. One in particular called the Natural History of Louisiana taught me how to really know the birds and look at them, and I got the opportunity to do a research experience with someone who became my PI, my principal investigator at Tulane University in New Orleans. And that research involved going to Australia and studying birds in their natural habitat. And so it was a really wonderful NSF-funded program. So if anyone is listening to this who is involved in making these decisions, please keep funding the International Research Experience for Students, also called an IRES, because it's an absolutely transformational program, as are all REUs, and I highly recommend that anyone does one if they have the chance. But this program basically taught me how to prepare research, how to write a proposal, how to come up with an experimental design. And not only did we go down and do the research, but we had to do the follow-through also. So then we had to write it up, we had to get feedback on it, learn how to make figures and do analyses and eventually present it somewhere in the form of a poster. So that really was how I got my first experience in science, and from then on I was sort of just grabbing at things and taking as much as I could, and so my interests were mostly ecological, I guess. I was doing a lot of field work. After I left the university, I was working for different agencies like the Bureau of Land Management, volunteering with the National Park Service, working at different research stations. And I realized that while I loved being outside and seeing the patterns in nature, observing what animals do or how plants are distributed or anything, I wanted sort of a deeper understanding of what's going on. And I really wanted to learn genetics and learn how to actually get genetic data, how to work with it, how to answer questions based on it. I never wanted to abandon, the field work, but I wanted to bring the lab to the field, sort of. So that's where I see myself right now, trying to situate myself at the intersection of, emerging genomic technologies and good old-fashioned hustling around in the dirt.
Amazing. So you said you did this research in undergrad, which I always really value experiences like that. They're really important. And then you worked around for a while. So how did you decide what university you were going to go to for grad school? And how did you pick an advisor and a program and a project? Because those are all really hard things to do. Yeah.
So while I was working, I had stayed at Tulane, my undergraduate institution, for an extra year to do a master's, thinking that I didn't want to do a PhD right away, even though I knew that was something I wanted in the end because I wanted to make research a career. I didn't want to do it right away, so I got the master's thinking, all right, I will work on this for a little bit, and maybe having this master's will help me get jobs or help me get paid a little more for those jobs, and it's debatable whether that occurred or not. But I did work for 3 years, and every year when, the fall would roll around, I would think, should I apply to grad school this fall? Nah. Because I liked what I was doing and I wasn't ready to commit to something. For me, going to grad school felt like I was going to be marrying my field. I was going to be making a very long commitment, possibly for the rest of my life, to a discipline of biology when I loved working in all of it. I hadn't thought of anything in particular that was more interesting than anything else. But I started, to really think, if I don't learn how to work with genomic data in a PhD, I never will. No one is going to teach me that for free. So if I don't get my hands on some of these techniques now, I won't be able to use them ever, probably. Or it will be much harder to learn them, and it will be at my own expense. So I thought about what I wanted to do in the future and how I would get there, even if it meant not doing what I most immediately wanted to do. And I decided, a lot of my friends were starting to go back to grad school. People who were younger than me were going to get their PhDs, and I was like, okay, maybe it's time. And so I decided to apply when a friend of mine who is a couple years younger was working on his GRFP application. And I was like, yeah, I should think about that. And then I look at it, I look at the deadline, it's in like a month, and I'm like, I should really do this.
Oh, it's so stressful.
So I just kicked into gear, sort of, and decided it's time to write a GRFP immediately and start finding some professors. I may have started a little bit late. I wouldn't recommend anyone starts 4 months or less before the deadline.
No.
But I had some ideas already because, I had flirted with it before. So I basically thought first, where do I want to live for 5 years? And Austin here was actually my top choice for that, so I'm glad it worked out. Some other places were like Florida, California, Washington, Michigan, mostly big state schools in those places I was interested in, because I had been at a small sort of liberal artsy type private school, and I really wanted the like major research university experience where, you've got 500 different labs and people are blowing things up over here and have a greenhouse over here, and it's just like an endless, research cornucopia. And all of the people I applied to, so the first thing was where do I want to live, and the second was who's there that I want to work with.
Yeah.
And all the people I applied to work under were bird people who did ornithology, many of them doing genomic techniques with them and stuff, except for the person I work for now, Justin Havard. And the reason he was different is because he was doing what he called mitonuclear ecology, basically the implications of these interactions between mitochondria and nucleus, and in a very integrative framework. So looking from the most small things you can get, these biophysical interactions of atoms on like such a small level, integrating that up to the fitness consequences for organisms and entire species. And I thought that was really cool, and I wanted to be able to do both the big and the small and see sort of the whole picture of evolution or of an ecological niche or something. And I had sort of heard about this idea of mitonuclear ecology broadly from someone named Jeff Hill, who's kind of the godfather of the field in a way. He's written a book now called Mitonuclear Ecology, so once you've got a book, you're a real discipline. And I met him at an ornithology conference. He's a very famous person from the literature on bird coloration, studying like the sexual signals of birds, what they do, what kind of information they convey, and how females make choices based on that, or other males and such. And that was the kind of work I had done for my master's. So I met him, we sat down for lunch. I wanted to talk to him about some of my ideas for this review I was writing, and instead he launched into like an hour of talking about mitochondria and what they do. And all of it went over my head.
Yeah.
And I was like, what are we talking about here? But I could kind of see what he was trying to get at, but I thought he was crazy. And he had the idea that when birds use red coloration as a signal, that they might actually be signaling their mitochondrial function, and that would then reflect how well their nuclear genes work with their mitochondrial genes. And since that time, he and his lab have demonstrated that we found that the enzyme that turns birds red basically works in the mitochondria, and his lab showed that the amount of red you get is related to the function of mitochondria. So he was right, and he had me go read a book called Power, Sex, Suicide, Mitochondria and the Meaning of Life by Nick Lane.
It's a title.
It's a great title, and I was sold on the title. And so I left that conference, read the book, and it totally opened my eyes to mitochondria. It made me start thinking about things in a more integrative way, and made me just sort of put these ideas in the back of my mind, on the back burner, kind of chewing them over. Like, I had no idea how I would ever apply this stuff or really work with it, but I knew that it was relevant now. And so when I saw Justin's lab working with mitonuclear ecology, I thought maybe I could do some stuff with birds, but through his framework instead of through like a bird-centered framework.
Yeah.
I still wanted to work with birds. That's my skill set. When you know how to catch an animal, that helps you get started.
Yep.
And Justin actually had been at Auburn where Jeff Hill was teaching, and I had known him and probably absorbed some ideas from him. So they were kindred spirits, and that kind of helped because I had thought about joining Jeff's lab also, but it wasn't quite right for me. So getting to work with someone who had worked with him was perfect.
Amazing. And I like that you use the word integrative to describe your research because, yeah, it does have all of these layers to it, but also, fun fact, Erik and I actually met one another, 2 months ago now at a workshop where we were talking about an entirely different kind of research. So can you say a little bit more about why integrative kinds of research are interesting to you and why were you at Succinct?
Yeah, so, well, let's start there. So Cheryl and I met at the Graduate Leaders in Socio-Environmental Synthesis workshop from the National Socio-Environmental Synthesis Center. And it's a mouthful. That's why we call it Succinct. But I was there because I basically don't want to be put in a box. And I'm not going to say that you can make connections between everything you do, but maybe you don't have to. You know, we are multifaceted people. We can have multiple interests. And for me, coming from a more ecological background, a more natural resources and conservation-oriented background, I've never lost interest in that. I'm very interested in doing conservation work in the future, and I don't know if that will be a component of my PhD right now, but it's not something I want to let go. So I wanted to learn how to integrate not just within biology, but across the natural and social sciences. I wanted to learn how to talk to people outside of my own field, outside of my own perspective, outside of my own discipline even, and to bring their insight together with mine and try to solve problems. And Lord knows we have a lot of socio-environmental problems. As we said in Succinct, all environmental problems are really social problems, right? So, you know, there's an environmental dimension to my work, but and there's a human health related dimension. Is there a social dimension? Maybe not right now, but I don't see this being the last research project I ever do, and I certainly want to stay involved in doing sort of integrated socio-environmental research.
Nice. So another one of the core ideas around the Explain Yourself series is this idea that anyone who wants to can and should become a scientist because we need lots of different kinds of people doing science. So is there anything in your background or like you as a multidimensional human being that you think has informed or affected your experiences as a scientist and like your experience within the scientific community?
Yeah, I mean, so I'm the only person in my family who works in science. My mom got a degree in biology, but then went into business. And for me, I was certainly able to stick with it because I had good support. And you need that kind of support. And I think it would be great if we could, you know, broaden the horizons of people who are influencing their children to see that like anyone from any background can do this. You don't have to like focus on just the immediate return of something that's more practical always. I mean, but there's obviously an economic dimension to that too. And I was privileged to have a firm economic background to go into something that could be a little risky. If I hadn't been exposed to the outdoors, I also wouldn't be here. So, I mean, I think we should really take students out in nature more, give them those kind of experiences that I had. And, you know, I've tried to do some volunteering in museums and zoos, get people excited about the natural world. And, yeah, those are the sort of experiences that really did it for me. It wasn't really so much seeing people in lab coats and thinking like, oh, that's cool, they're doing like groundbreaking research, you know, that's going to change the world. It was more like, I like being outside and I like animals, and it sort of snowballed out from there.
All right. Nice. And the other thing I like to ask everyone, so we are scientists, we identify as scientists, and that's how we present ourselves at the beginning of this, but you are a scientist and also other things. So is there anything about you or like hobbies or other interests you have that you think would surprise people to know about you, that you are a scientist and also?
Well, I'm also a poet.
Really?
Amateur, but have been published. So I think people should know that, you know, being a scientist doesn't mean only working with that sort of analytical part of your brain. Science is actually a very creative endeavor, and a lot of scientists have creative outlets.
That's amazing, and I'm going to geek out a little bit about that because, as viewers may or may not remember, I also have a liberal arts background and an English degree. So where has your poetry been published?
So it has been published in sort of a small magazine that I can't even remember the name of because it was in high school.
Okay.
Through basically a competition there. And then I've published in 2 other zines. One was from Tulane, basically. It was the Tulane Literary Review, I believe. And another one was a publication of a friend called Fish Like Sharks. It's fantastic. You should pick it up. And then I've also submitted something to Orion magazine, which deals with the intersection of people, nature, place, and it's one of my favorite things to read because you've got your science and your art coming together in that magazine. I love it.
So how did you get into writing poetry?
Through a great teacher, like a lot of things. Through a creative writing class in high school, we had a couple electives you could take for English, and most of my friends were doing AP literature, and I was like, I have too many APs, so I'll take creative writing instead. And you know, that got me started with writing maybe 10 or 15 poems as assignments for that class, but it doesn't really feel like an assignment once you realize you like doing it a lot. A lot of them were sort of sprawling, wordy rant type poems.
As
and then
teenage poetry often is.
Absolutely. And then you learn to condense and to sort of make it accessible to other people also. You know, what is a common experience someone could share rather than just your own internal monologue?
Amazing. Is there anything else you would like people to know about you or about science or about getting into science?
Yeah. I think it's really important for scientists not to view themselves as people who have to stay objective. I think there is this sort of myth that scientists, you know, are supposed to just look at the world dispassionately and evaluate the facts and not have opinions on things. And I think that's bogus, especially when scientists have expertise on so many issues that we face in society. But scientists also are trained to critically evaluate things and make decisions. And so you can apply that outside of science to the world around us. So I think scientists should speak up about social issues that they see. I certainly have, if anyone knows me or my online presence. But I've also been involved in a number of organizations that try to basically, you know, either improve people's lives or advocate against injustice in our society. And I think some people look at science and they don't see that going on, and they think this is not the place for me. They think, I care about social justice, maybe I'm from a minority background and I've had to deal with these issues and I don't see anyone speaking up for people like me in science. So I think it's really important that we do that, that we are not only inclusive, but we advocate for justice and not just sit on the sidelines. There's an organization that I've been working with called Science for the People, and I want to highlight that just because I think their work is really critical and very little known. This organization basically advocates against the misuse of science in our society. So we are trying to make sure that science is not used to harm anyone, that science is accessible to all people, and the benefits of science flow to all people. And this movement was rooted in the anti-war movement of the 60s, actually, in the anti-nuclear movement. That's how Science for the People began. And the idea was, we are spending billions of dollars and, you know, millions of person hours designing weapons of destruction, and just imagine what that effort could do if it was applied towards solving our problems. So this organization has had a resurgence in the last few years. We've started republishing the magazine, which was sort of our original flagship, and now chapters are being organized around the country. And we are basically trying to get scientists, STEM workers, science educators to think about themselves as parts of science as an enterprise that have leverage. Everyone who works in science has a little bit of leverage. You have control over what you research. You don't have control over how it's used, but you can advocate for it to be used one way or another. And you should think about the consequences of what you're putting out there before you publish it. So we have been active against, like, the use of science for biological determinism. We're trying to, this sort of resurgence of race science in our society thanks to genetic data. We have been trying to get tech companies to stop supplying technology that is used to break up families or do surveillance on people. We're advocating for a Green New Deal, for the application of science to fixing the climate crisis rather than coming up with new technologies for producing more fossil fuels. And one thing I would just like to highlight is that a chapter of Science for the People, or one of our working groups that I'm involved with, is called Science for Puerto Rico. And what this group has been trying to do is to basically help the island recover from Hurricane Maria by listening to what people there want, average people, not what the rich who are sort of prioritizing the island are asking for, and to help Puerto Rico become a sustainable green economy by investing in agroecology, investing in solar technology. And so in 2017, Science for the People sent a brigade down to Puerto Rico of a bunch of scientists and advocates, and they basically, went into communities and listened to what they needed and helped with various projects, whether that's working on a farm and trying to make, some sustainable agriculture so people can grow food and not be so dependent on food from the outside world on a small island, or installing solar panels to try to get the island off of fossil fuels. So we believe that science has a role to play in all these crises, including like the immediate crisis of responding from a hurricane and the long-term crises we face from climate change and other things.
That sounds like an amazing organization doing a lot of good work. So if you're interested, there will be links down in the description, and I'll also put like social media and stuff so people can learn more about you and what that organization does. So yeah, I didn't know about that before. Thank you. That's interesting. So yeah, thank you so much for talking with us, Erik. I always have fun talking with you and listening to what you think about, whether it's research or socio-environmental problems. So I appreciate you being on the show.
Thank you for talking with me. I enjoyed it so much.
All right, so don't forget, if you liked this video, don't forget to like it. If you didn't like this video, please share it with someone who would. And if you'd like to support the Roving Naturalist, remember to hit the subscribe button and the bell notification icon. You can also follow me on Twitter and Instagram. There are lots of scientists in this playlist, so maybe watch through the whole Explain Yourself playlist. I feel like as it grows, it becomes more and more valuable, so please give it a watch. And as always, thanks for watching, and I'll see you next time.
Bye.